Hi Year 10, today we're doing lesson 4 of the plants topic and we're looking at gas exchange in plants. And we're going to start by considering the organ in plants where gas exchange happens, and it's in the leaf. So a lot of today's work will focus on the structures within this leaf and how they help with efficient gas exchange. So just to get a sense of what we're looking at when we look at the diagrams, what we're going to do is look at a cross section through a leaf. So that would be if I take a slice through this way, a very thin slice. So we're going to see a very thin section and we're going to magnify it. So what we see when we look at an image like that under the microscope is something like this. This has been slightly simplified compared to what you'd see on a microscope. But it has all of the structures that you'd expect to see if we managed to get a little thin section of this leaf. Now hopefully some of this looks familiar to you because you will have met this at Key Stage 3, but we're going to go over first of all the key structures that you need to know, and then we're going to look at how gas exchange occurs and the special features that this leaf has in order to make gas exchange efficient. So we'll start with our structure labelling, and hopefully a lot of these words are familiar to you. So on the very top surface of the leaf we have a layer of a waxy substance. We call this the waxy cuticle, and that's there to reduce water loss from the leaf. So it reduces water loss. Now water loss is a process called transpiration, which we'll be looking at in a future lesson. The next layer down, this top layer of cells here, which are completely transparent, this is the upper epidermis. Epidermis essentially means skin or surface. Then we've got the most important layer in terms of the function of the leaf here, these sort of tall stacked cells. We say they're columnar, they're column shaped. This is the palisade layer, and this is the main site of photosynthesis. Oh, let's get that in the right place. Main site of photosynthesis. And because of that, the cells in this layer have lots of chloroplasts. Those are the green blobs that you can see here. There are chloroplasts in other cells, but this is the layer that has the most chloroplasts. So lots of chloroplasts. And that's so they can absorb the maximum amount of light and carry out lots of photosynthesis. Underneath that we have a similar section here. This is called the spongy layer. And it's called that because of these air spaces that sit within it. And the spongy layer is all about gas exchange. So those air spaces are linked to gas exchange, which is what we're going to go on to in a second. Then at the bottom, mirroring the upper epidermis, we've got a very similar layer here, the lower epidermis. And within this layer we've got these little pores in the bottom, which are called stoma, or the plural for that word is stomata. So if we have more than one, we call them stomata. And they are surrounded by cells called guard cells. And the guard cells are there to open and close the stomata. And the stomata themselves are there to allow gases in and out of the leaf. Okay, so those are all our main structures. We've also got some transport tissues over here. We've got the xylem, which we will look at also in more detail in a future lesson, but we have mentioned it already, and the phloem. And we find these in these structures here. They're essentially veins, and in the plant the proper name for a vein is a vascular bundle, but you can just call it a vein, that's okay. So we've got all these structures, but the main features that are important in terms of gas exchange are stomata and these air spaces. So the stomata are going to allow gases in and out, and for the plant it's really important that they can get carbon dioxide, CO2, in, because carbon dioxide is used for photosynthesis. So carbon dioxide comes in through the stomata and it will diffuse into cells out of these air spaces and get passed through the leaf, so that any cells that need carbon dioxide will have some. And then diffusing out of the stomata at the same time, there will be some oxygen, which is produced in photosynthesis, although some of it the plant will use for respiration, and there'll be some water vapour, or H2O, and then the state symbol G for gas, water vapour. And that water vapour the plant doesn't really want to lose, but it happens as a result of these stomata, and we will come back to that when we learn about transpiration. So these gases are moving in and out of the air spaces by diffusion, which we learnt about back in year 9. So this is movement of particles from an area of high concentration to an area of low concentration. And so in this case, the carbon dioxide has a higher concentration outside the leaf than inside, so it moves in, and the oxygen and the water vapour are the other way around. Their higher concentration is inside these air spaces compared to outside, so they move out. Okay, so let's consider what makes an exchange surface good, because essentially this is an exchange surface for carbon dioxide and oxygen. So what is it that makes an exchange surface good? And you should already be aware of some key features of specialised exchange surfaces, but let's go over them. So generally we can break this down into 4 key things. So number 1, all specialised exchange surfaces have a large surface area to volume ratio. You can write that like that, SA colon V, surface area to volume ratio. They also are thin. So being thin provides a short diffusion distance. So if whatever you're exchanging has less far to travel to get into the cells, then it's going to be more efficient, quicker essentially. Number 3, all exchange surfaces have to be moist. So there's got to be some moisture on the surface of those cells, and that's really important because for substances to diffuse into the cells, they need to be dissolved in water. So substances need to dissolve in water to diffuse into cells. And then the 4th feature isn't found in all exchange surfaces, but it is key for animals. But you'll see why in a second. It's not really a feature in the plants. So number 4 is that exchange surfaces often have a good blood supply or ventilation over the surface. Ventilation is active movement of air, so like when you breathe in and out, that's ventilation. And this is really useful because it will maintain a steep concentration gradient. So what that means is if we are moving a fluid or air past our exchange surface, we're going to be taking things away from that surface, and it causes the difference between the blood or the air and the cells, the difference in concentration, to be greater. And the greater that difference, the faster the rate of diffusion. But this is something plants don't do. They don't have a blood supply. They do have transport tissues, but it's not quite the same. It's passive. And they don't breathe. They don't ventilate these air spaces. This is passive diffusion in and out. So don't ever talk about plants breathing. So let's think about how these specialised features are achieved in the plant. So the large surface area, well, that's essentially, if we go back to our leaf, achieved by being big and flat. I mean, this is a relatively small leaf, but if you've studied rainforests in geography, for example, you look at the leaves you get there, they are enormous. And by being large and flat, we've got this large surface area. But we also have an extra large surface area if we have a look at our leaf. Let me do this in red. Within the leaf, these air spaces allow the carbon dioxide rich air to be in contact with more cell surfaces than they would if it was just a flat surface on the outside. So the air spaces in the leaf provide this much bigger surface area for gas exchange as well. And every single airspace is providing this, so we could show that on our diagrams like this. There's a couple more there too. So for the plant, the large surface area is achieved by both leaves being flat and large, but also by having these air spaces in contact with lots of cell surfaces. And this essentially is the equivalent to having a folded surface like we see in the small intestine, for example. Okay, thin, well, a leaf, if we look at that, a leaf is very thin. And what that means is the distance the gases have to diffuse to get to the cells is really short. So leaves are very thin. So diffusion distance between air spaces and cells is short, and that means it's going to be able to happen quickly. Sorry, I went off the edge there. And then in terms of moist surfaces, so the cell surfaces themselves are moist. They have water on the outside, and that means that the carbon dioxide in these air spaces can dissolve in that water on the surface, and then it can diffuse into the cells. So cell surfaces are moist, so that carbon dioxide in the air spaces can dissolve in the water and diffuse into the cells. So that's how the leaves are providing a specialised exchange surface for the plant for gas exchange. Now, there are other structures that you've already met in previous topics that have similar features to this, and so we can relate these together. So other structures with similar features for exchange, or for efficient exchange. So you will have learnt about, when you did the digestive system, you will have learnt about villi. So villi are these finger-like projections on the surface of the small intestine, and they provide a much larger surface area. They are also only one cell thick. That's my one layer of cells there. So the diffusion distance is very short. And they also have a very good blood supply. So there's lots of capillaries within the villi that are supplying blood, but also are taking away the products of digestion that will be diffusing in. So that's the villi in the small intestine. So we've got a large surface area, we've got a thin wall, and so it's a short diffusion distance, and we've got a good blood supply. And also that surface will be moist so that we can dissolve the products of digestion in the water so they can diffuse through. And another one that you will have met in your last topic is the alveoli in the lungs. So these are the air spaces within your lungs, so performing a very similar function to the leaf. This is where gas exchange will happen in your body. And again, we've got a layer around the alveolus that is one cell thick. We've got this shape where there's literally millions of these spherical, almost like bubbles, providing a much larger surface area, or surface area to volume ratio. And we also have, in contact with the alveoli, blood vessels, these capillaries. There's a really rich capillary network. So every alveolus has capillaries around it. So again, we're able to maintain a steep concentration gradient. The alveoli also have active ventilation. We've got breathing in and out, moving the air through here as well in order to maintain that concentration gradient. And there are some other surfaces you could come across where you might have to apply your knowledge. So for example, gills in fish or in amphibians. But all of these exchange surfaces have the same features. So I'll just zoom that out so you can see the whole sheet online, hopefully. Almost. And so this is a summary for gas exchange in plants.